joining us from Spain is Professor Alfonso Martínez Arias, who is a research professor at the Catalan Institution for Research and Advanced Studies and at Pompeu Fabra University. He is the author of the book The Master Builder: How the New Science of the Cell is Rewriting the Story of Life, and he is co-author of the book Principles of Development. And finally, Alfonso is an advocate for raising standards in research, particularly involving stem cell-based embryo models.
I never met Professor Edwards, although I met some of his students. And as I will refer with a reason at the end, for over 20 years, I worked in a building that is next to the Marshall Building in Downing Street, where a lot of these very important developments took place. As a developmental biologist, for many years, I took IVF as a technique, something that allowed infertile women to realize the dream and to barren couples to have descendancy. However, as my interest in mammalian and developmental biology and human embryos grew, I started to understand something that sometimes is missing in the story of IVF. And this is that without developmental biology, IVF would not exist. And it surprises me to this day that textbooks of developmental biology and developmental biology courses do not make more of an emphasis on the fundamental roles that are played in the history of IVF. If one looks at the time span over which IVF was developed, one can see two very clear periods. One that goes from 1961 to 1971, which is, to my liking, pure developmental biology. And then the second part, which relies on an interaction, again, between developmental biology and reproductive medicine, in particular endocrinology and gynecology. And it seems to me that Bob Edwards' work, it's a very good example and an important one of the great benefits that follow from the collusion and the interaction between developmental and reproductive biology, which I think it's missing today. And I think Geraldine has made the point today that there is a lack of funding in this area. And I would say that part of the problem comes from not acknowledging that IVF is not only a part of clinical research, but it's also a part of fundamental research and in particular developmental biology. During that first period of the history of IVF, as Barry Babister has explained to us, the most exciting moment was the time in which a sperm managed to fertilize an egg in the dish. Today, we can see this event with great beauty, and this is part of some textbooks of developmental biology. Here at the bottom, you can see the moment in which the paternal DNA, it meets the maternal DNA and it starts mixing. And here you can see the chromosomes in the dance that they engage when they are starting to mix and become part of the zygote of the future embryo. This, in a way, would be the modern version of the paper by Edwards, Babister, and Steptoe that was referred to at the beginning of the meeting. And there you have a sentence from the book, A Matter of Life, which I think Nick will discuss later. But it holds, in a way, the amazing fact, and that's what Barry was referring to before. And that was the first time that the sperm was seen inside an egg in the dish. To me, a most important event is what ensued, because as I often ask the students, how would you know that a sperm has fertilized an egg? Well, they take some time to realize that what happens is that the egg starts dividing and it starts producing first the morula, which is the way the embryo starts making itself by amplifying the number of cells, and then eventually the blastocyst. Here in the middle from one of the papers of the original paper, this is the human blastocyst grown in culture, which is a Steptoe, Edwards, and Purdy. Here you have a modern picture of a human blastocyst. This is actually the embryo cells are sitting here surrounded by the trophoectoderm, as it's called, that will give rise to the placenta. To me, this probably was, as a developmental biologist, would have been the most exciting moment Nobody had seen a human embryo in these very early stages, and much less had seen the emergence of these blastocysts, which always happened in secret inside the uterus. It is this study, these methods that they developed that have allowed our vision today of the early development of the human embryo. This is taken again from the book A Matter of Life, where Edwards states, I was thrilled when I observed the embryos we had incubated divide into 2 cells, 4 cells, 8 cells, and more, each with their own nucleus. And he is thrilled, he expresses his thrill at the vision of this, as many of us do every time that we confront this. And as I said, I want to emphasize this is now a very important part of developmental biology, and it is the methods that they developed in the early 70s that allow us to have these views and to move forward. This technology also allowed us to develop embryonic stem cells, which have become a very powerful tool in the tackling disease and also cures for regenerative medicine. And as I said at the beginning, it surprises me that we do not recognize that behind all these very important branches of research, it is this very pioneering work that branches off into IVF, but laid the foundations for what we do today in human biology. And of course, this kind of work has also led to the stem cell embryo models that Sarah has referred to at the beginning, and which allow us to replicate in the plate now the blastocysts. And all this comes from the stem cells that were derived from those structures and the ability to grow them in the dish. This work is very important today, and it's been the reading of the life, the work, and the thoughts of Bob Edwards that is guiding many of us in realizing that research at the same time that provides basic knowledge, it has to be done within ethical frameworks that are very important to bring about the public and the funding bodies. And I don't need to tell this audience, and certainly many of the people that are here with me this evening, that Bob was a great advocate, and from the beginning appreciated the value of carrying the ethics of the field, laying down the ground foundations of it as he did, because at that time nothing existed. And we are taking on board his important admonishment of carrying together the ethical aspects of the field with the research aspect of the field. The first part, as I said, ends up with the generation of the blastocyst, and then it looks as if it's Elysian. If you read the history and you follow the writings of the people around Edwards, you might have thought that having been able to fertilize an egg and grow it in the dish, implanting it would just follow suit, that a baby would appear a few months later, but that wasn't the case. And it is because this part required an interaction between gynecology, endocrinology, reproductive biology, and this basic research in the biological sciences. And this took a long time, and it was more, as many clinical treatments are, a little bit of trial and error. And in the end, it was very surprising that despite many treatments that they tried, they ended up doing it, as it's been referred before, the natural way, with 1 egg at a time, 1 sperm at a time. It is this part of the whole procedure that today, and I agree with what has been said before, it needs a lot more research, and it needs the acknowledgement that this is not just a clinical treatment, but part of the biology of reproduction that needs to be attended in funding. So it is that convergence of developmental and reproductive biology that will lead to a better understanding of IVF and to an increase in the yields and also in the health of both the women and the children. I want to finish by saying something that it's very obvious, and it was very obvious to me the minute I started thinking and reading and learning and talking to people about this. This is the Department of Genetics, where I worked for over 20 years, and this is the Marshall Building. And I never understood that in a city that it's so proud of all the biology that it's developed, particularly the DNA, the genetic code, I never understood why it took so long The plaque was unveiled in 2012 to acknowledge the fantastic and important and fundamental contribution, not just to science, but also to life, as it's been said before. It's something that escapes me. Perhaps it is because, as it's been said, and it's been said a number of times in many places, fertility is not still considered a big priority in many of the problems of the world, when actually it is. So I am a great advocate for what Geraldine has said before, that we need to provide more research funds to this field. And I think that what should be emphasized is this is not just the improvement of a technique, but is the result of the crossroad between basic biology and more clinical aspects of reproductive medicine. And I think we need to emphasize that the fundamentals of this technique lie in developmental biology, which is a major part of biology. And perhaps from that side, and acknowledging the contributions that Edward's made to this field, we can convince the bodies to provide funding.